Welcome back. Hope you had a nice holiday on Monday. We are on to the second part, chapter on evaluating causal hypotheses. Um, this is possibly the longest lecture in this course. Try to keep it as close to 50 minutes as possible. But we're covering two other types of designs that we can use to evaluate causal hypotheses. So let's jump right into it. I won't dawdle. So for the first model, we are going to use a study of schizophrenia and heredity. So, schizophrenia, prevalent psychiatric disorder. I'm almost certain all of you have heard of it, right? It affects about 1% of the population. Um, it's diagnosed using sort of both positive and negative symptoms. So, positive symptoms are probably the ones that are more commonly in the popular imagination associated with schizophrenia, like hallucinations, delusional beliefs, and such. There are also negative symptoms, which are sort of like withdrawn aspects, sort of like lack of emotional response, even catatonia, right? Just total um, sort of silence, non-moving. Um, Schizophrenic episodes typically manifest in the late teens or early 20s, um, is when it starts to kick in people. So the issue at interest here is whether schizophrenia is a biological disorder, right? It's a mental illness, right? But what is the biological basis for that, if any, right? Um, among the various hypotheses, the dopamine hypothesis, which says it's an excess of dopamine being released, right? It's a neurotransmitter. Um, and experimental tests of that hypothesis sort of produce some mixed results. Um, but one good reason to think that schizophrenia could be biological would be if it is passed on through genes, right? Um, and we do see in adoption studies that schizophrenic children of parents with schizophrenia do have greater rates of schizophrenia as well. The reason we're focusing on subjects who have been adopted is that you want to eliminate environmental factors, right? So the question is it purely genetic, right? Um, in which case, the schizophrenic child is not raised by the schizophrenic parent. That seems to eliminate that possibility of purely environment. Another way of addressing this issue um, is to use twins. So monozygotic twins are what we call identical twins, right? They come from a fertilized egg that splits, 100% genetically identical. Dizygotic twins, what we call fraternal twins, so they come from different eggs that are fertilized from different sperm, and they just are both gestate right in the womb at the same time and are born at the same time. So typically they'll share maybe up to 50% genetic material. So this is sort of classic method now, the twin method, and there have been a number of studies using this method. So the researchers of the study we're going to be evaluating sort of did a review uh, of a number of studies, right? So here's how it would go down, right? So you look through databases and try to find um, twins, at least one member of the pair at least has schizophrenia, right? And then you want to see if the other member of the pair has schizophrenia as well. So if both twins have schizophrenia, then it's called a concordant pair. Um, and if it turns out that the other twin doesn't have schizophrenia, we call it a discordant pair. So the idea is that if you're twins, there's going to be no difference in environmental factors, right? You will have been raised in the same home together. So it's about as close as you could get as far as environment. Um, and so if there are systematic differences between identical twins, right, MZ twins, and the fraternal twins, the DZ twins, um, the only reason for those differences would be because, presumably, the monozygotic twins have greater genetic similarity, right? So if basically if the twins that are closer genetically also tend to be more frequently concordant, right? So if one has schizophrenia, the other has schizophrenia, that is suggestive of a genetic basis for schizophrenia. Um, so there's two different ways to calculate concordance rates. It's not super important for us, right? Um, Pairwise is the easiest to make sense of. So that's just the percentage of the sample of either the MZ or the DZ twins that are concordant or discordant. So, for example, if 10 of, say there's a sample of 50 MZ pairs and 10 of them are concordant, then that's a 20% concordance rate. So that should make sense. Um, the proband version places more weight on pairs where each one was assessed to have schizophrenia independently. And in the study we're going to see, we'll use proband rates. Don't worry about it. Just think of it again as basically like what percentage, right, are concordant pairs. That's the main thing. So these twin studies have developed through the years, right? So early on, twin studies found very high concordance rates for MZ twins compared to DZ twins, right? So 86% versus 14%, which seemed to be, very strong support for a genetic basis for schizophrenia. But as the sampling methods got a bit more sophisticated, you definitely got uh, less impressive concordance rate, but still significant, right? So again, this study that we're going to look at combines the results of 5 recent studies and find concordance rates among the MZ twins between 79% and 43%, and then the DZ twins anywhere from 17% to 0%. So just eyeballing it, right? Pretty significant differences. So, but we'll do it uh, the right way. Well, so you pool all these together, right? From these different studies, all the data, and so they use criteria. So the DSM is a book that's just sort of it's a standardized way to diagnose mental illnesses, right? So and there's DSM is one way to do these diagnoses. There's another one called the ICD, um, and so in this study they give the data for both ways of diagnosing, right? These twins. But basically, right, so MZ twins, 50%, right, were concordant, again, meaning that both twins had schizophrenia, um, if one did, and 4% for the DZ twins. So pretty different, right? And then if you're using ICD criteria, it's 42% and 4%. So still pretty different. So um, clearly, like significantly higher concordance rates among the MZ twins, taken as evidence supporting the hypothesis that schizophrenia is in fact, right, a hereditary condition. Here's right, some of the actual data. So we can see the 1, 2, 3, 4, 5 different studies, right? One in Denmark, one in Finland, Germany, UK, Japan. Which diagnostic, how they found the people, right? Through hospital admissions or through the population, like birth records, the diagnostic criteria that were used in each case, and then the data, right, that they found. So in the Denmark study, right, 44% concordance rate among the MZ twins compared to 11%, and on down the list, right? And then they have them pooled there at the bottom. So according to DSM, right, if you pool all the data, 50% concordance rate among MZ twins compared to 4% among DZ. And here's the combined data, right? So again, among MZ pairs, 50% concordance rate compared to 4% for DZ, according to the DSM. And on the right, you have using the ICD version of way of diagnosing people, slightly fewer concordant pairs among MZ, 42%, still 4% among DZ, and still difference. So why do we bring up this study? So this study is an example of a kind of model that we're interested in. So it's called a prospective design. Um, so as we saw with the rats, right, and the saccharin bladder cancer studies, where we were able to just randomly assign subjects to experimental and control groups, right? You could just randomly sample from the population and then randomly just split them into groups. Here they don't do that, right? They find subjects that already have the suspected causal factor, and they find subjects that don't, right? In this case, they're sort of selected by nature, right? They might have selected themselves, right? If you're looking at smokers, right? People choose to inflict the causal factor on themselves, right? They choose to smoke. Twins don't choose to be twins here, right? Here the causal factor, right, is genetic similarity, right? But in either case, in a prospective design, they sort of find the populations, right, where they all have the causal factor and they all don't, and then they just sample from those populations that are already out there. So instead of randomly assigning people, right, a group to all get the causal factor and one group to not get the causal factor, they just go out there in the world and find groups that are already like that and just sample from them. So the strategy, right, so it's a little less random, right? So you want to try to find two groups that are as similar as possible in every way, except for the suspected causal. And then the idea is, right, if the two samples exhibit a statistically significant difference in the frequency of the effect, this will be evidence for the causal hypothesis, right? So again, it mimics, if you go remember back sort of 2 lectures, right, but on the basic idea of how do you make an inference back to a causal claim, right? The idea is like, well, if you had a possible right world, in a world where everybody had the cause, in a world where everybody didn't have the cause, you should see higher frequency of the effect in the world where everybody had the cause. Well, they sort of recreate this here, you know, as well, just by sampling from groups that all have the cause, don't have the cause. Right. So here's kind of what I just said, right? So here's a real population, and some of them have the causal variable, right? Some of the people in the population are not going to, so you just sort of create your groups from those, create your sample from those groups that already exist. So you've got your X group, they all have the causal factor. You got your K group, none of them have the causal factor. And you see down in the real world population, right, some percentage of people with a causal factor will have the effect. And in the non-C group, some percentage will have the effect as well. But if that percentage is lower, right, if that probability is lower in the real population, that means that there is a causal relationship, right? So if we find a different frequency in our sample, we can estimate backwards, right, and say that, given a margin of error, we can estimate, as long as there's no overlap, then there is a in the populations, which means there is a causal relationship. I went over that fairly quickly, but again, it's no different than what we saw 2 chapters ago, right? And again, with the mice, that basic part of it stays the same, right? The basic idea of how we determine that there's a causal relationship. So, okay, so you can see, in a way, a prospective design looks a little bit more like a test of a correlation than it does a randomized experimental design, right? But here's the difference. In a prospective design, when at the beginning the effect is not present yet, right? You're looking at 2 groups that have the cause, one that has a causal factor, one that doesn't, and seeing if the effect pops up. This is going to be more reliable than a one-shot survey in which you sort of ask people after the fact whether they had the causal factor, right? Right. So if you go asking people with lung cancer, how much did they used to smoke? It's not going to be very reliable. They're not going to remember it very well, and the fact that they have cancer will very likely color their perception of that past behavior. Now, you may be asking, what's perspective about this? They didn't actually do that, right? They didn't identify these twins at birth and then wait to see if they developed schizophrenia, right? They went through hospital records, found people with schizophrenia, and then looked backwards. More on this later. We'll explain why this is still perspective in a moment. Okay, so one important thing here, right, is this is not as random as the random experimental design, right? We have not sort of randomly chosen from the population, right? We're looking at people um, with specific causal and non-causal factors, and then we're not sort of splitting them into groups randomly. So how do we approximate a random sampling model with a prospective design? Um, and this is always an issue. This is a problem for prospective design. Why they are if you get to choose the random experimental design, right? But sometimes that's not available. So when you're doing a prospective study instead, you have to be very careful to do your best to approximate randomness, right? So um, this is the whole reason we're using twins, right? It's supposed to control for a whole host of environmental factors that might be different that could turn out to be causes of schizophrenia, right? So the idea is, okay, these twins, they grew up in the same house, right? So if one develops schizophrenia and the other doesn't, um, it shouldn't be because they were in a different environment, right? Um, so, right, so this is why. So if the MZ twins exhibit more concordance than the DZ twins, um, we shouldn't blame it on the environment, right? Because both are twins, uh, both are sets of twins, both each set growing up in the same household, right? And presumably there's no systematic difference. Well, this will be an issue, right? Is there a systematic difference between MZ household, right? Households that have monozygotic twins in them and households that have dizygotic twins. Um, right. This is sort of the next point. Like being an identical twin is a pretty different situation than having a fraternal twin, right? So if you um, like people know you're identical twins, right? When you walk around together, people see it. If you're dizygotic twins, right, you might be a different sex, or at any rate, you're not going to look identical, right? So you walk around and people just say, "Oh, brothers, you seem pretty close in age," but it's a pretty different situation to sort of have a genetic duplicate growing up in the same household. So who knows what sort of effect that could have on mental health, right? That's a potential problem. So this is sort of you can think about sort of the same data and just think, okay, if there is some environmental factor that's different in MZ twins and DZ twins, then it might totally be that the concordance rate is due to that, right? Um, how do you control for that? So um, control for that. I realize I just used that term, but I don't think we've technically introduced it yet, right? So you may have heard of this term um, or as controlling for other variables. So um, the idea is we want the causal factor we're interested in, right, is the genetic identity, right, or genetic similarity. And we want to try to rule out other potential causal factors, right, other differences between those two groups, right, besides their genetic identity or lack thereof. Um, so the way we rule out those potential causal factors is we control for them. So to control for that particular um, issue, right, of having a sort of identical person to you walking around, um, that's very difficult to control for. You'd have to somehow try to find um, monozygotic twins that just didn't, as a result of that, I like looking identical, didn't have any sort of different sort of experience growing up, right? Um, Similar parental treatment, similar treatment in school, and so on, right? How do you do that, right? I mean, maybe if the um, monozygotic twins went to different schools and sort of didn't so different people didn't see them together all the time, didn't treat them as sort of odd for being identical, but then now you've got a difference you've introduced, they go to schools and so on. So it's tricky, right? Maybe impossible, um, but in principle, I get right. It's not in principle possible, even though it sounds very, very difficult. What I'm saying, um, you could control for every single other possible causal. Um, sometimes when it's really difficult to control for that, one way to approach it is to sort of like flip it and treat the potential alternative cause as the causal variable, right? And then control for the hypothesized cause and see if you find evidence of an effect, right? And if you don't, then you can say, okay, I guess it's not a causal factor, and we can sort of forget about it, and now we can go do our regular study, right? If that makes you couldn't do that with this particular study because you'd have to control for genetic identity. Remember, you're sort of flipping it and saying, okay, we'll make um, right the potential alternate cause, right? So the odd treatment that identical twins get, make that the causal variable and see if there is an effect, right? Um, but you can't control to control for genetic identity. You'd have to get rid of the MZ. So it wouldn't work for this, but it can work for other types of studies. So I mean, one way to right control for variables is if you can think of an alternative causal factor, right? Um, then you can look through your sample, and if anybody has that alternative causal factor, you can just pull them out, right? You can eliminate them from the study. Um, that would work, but it's also going to reduce your sample size, and you would like to keep your sample as big as possible, right? Um, they did some of this in the twin study. So some of the pairs, right? One of the twins died young before any sort of schizophrenia um, screening could be done. So those got removed, right? They also removed dizygotic twins if they're of different sexes or genders, um, because men and women get treated differently in this world, right? And the point of the twins was like to have them as similar as possible, right? So they left those out as well. Um, Sample size getting smaller and smaller, right? Uh, you want to keep your sample size bigger instead of controlling by eliminating subjects from the study. You can keep those subjects so long as you match them across the two groups, right? So um, if you got one part in the experimental group that has a potential alternative causal factor, we'll just have make sure you have one of those in the experimental group as well, right? Um, that wouldn't work for this particular one of like eliminating the DZ twins of different sexes because none of the monozygotic twins will be of different sexes, right? You can't match that, but there might be other um, variables that you could match, right? So if you want what people from countries, right? So if you got multiple countries and you got some twins from France in the control group, make sure you got some twins from France in the experimental group, and so on. Make sure that if France is a causal factor, well, that's balanced out. So like if you still see an effect, it couldn't have been being in France, the reason, right? Um, okay. One issue with that matching is that there might be interaction effects, right? So maybe being from France on its own isn't a causal factor, and it only becomes a causal factor when it interacts with your being a monozygotic twin or something, right? And then matching's not going to help, right? Because even though you've got the same, again, potential alternative causal factor in each group, it might be that that factor only becomes sort of activated when it interacts with the other factor, right? So these are all just things they have to think about when they do these studies, and when you evaluate it, consider it too. We're going to see these when you look at these valuations, you're going to get you've probably already read the book, but you're going to get a little freaked out. I'm not going to expect you to do this kind of detail at all on the exam. Right? We're going to keep it pretty simple, but just for your enrichment and understanding this stuff, it is useful that they give very detailed, evaluations. But don't look at those and think, oh my God, I'm going to have to do that. You won't. Okay, moving on. Trying to get through this in a reasonable amount of time. Okay, so perspective designs. It's kind of clear at this point, right, why a randomized experimental design is going to be better evidence for causal hypothesis. Um, really preserve the randomness of the sampling much better, right? um, by randomly assigning individuals to experimental control, right? You're confident there's no systematic differences between the two groups. If you do see a difference, it must be the causal factor. You know, statistically possible that even in a random, um, right, even when you split them up randomly, that one group could end up with a greater proportion of an alternative causal factor that you didn't know about, but it's just statistically unlikely, right? Um, Whereas in a prospective design, you don't get to randomly assign them. They've assigned themselves, or nature has assigned them. And so you have to do a lot of work, right, to determine whether there is some other causal factor at work here. Okay, so here is the evaluation they give you in the book of the twin study of schizophrenia. Don't freak out. You don't have to do anything this detailed in the exam, but let's walk through it, right? We can at least understand it. So step one, real world population and causal hypothesis. So the population of interest is all adults, anyone who might have schizophrenia. Okay? Population sampled consisted entirely of pairs of MZ and DZ twins, Europe and Japan, born between 1930, which is where the earliest data from these studies, in 1972, which was the latest data. Again, this was taken from like hospital records and birth records, so it spans a wide time period. Um, and yeah, again, so they found them through national birth rate data banks, admittance records in psychiatric hospitals, and they matched those against the twins' birth records. So the causal variable is the genetic material passed along, right? And the effect variable is a schizophrenia diagnosis using either the DSM-3 or the ICD. And the causal hypothesis is that genetic material is a positive causal factor for schizophrenia. All right, in these particular twin pairs. Okay, so what is the data? We've already seen the data, right? So, but the sample was 180 MZ pairs, 174 DZ pairs. Um, Again, at least one member had been diagnosed with schizophrenia. Um, and then by the end of the study, about 40% of the MZ pairs were concordant, right? Turned out that both twins did have schizophrenia when they sort of investigated some more. And then using DSM-3, sorry, so 40% concordant, 50% using DSM-3, 42%. I see, that's an average, okay, of the DSM and the ICD. That's what they're doing there. Look through this, but then I just got confused for a second. And then the DZ pair is much lower, right? 4%. Okay. Okay, so step three, the design of the experiment. Here you just pick out what sort of design are we dealing with? Is this a randomized experimental model or is this something else? This one's clearly a prospective model. We've been talking about this, right? It's not experimental because there's no random division into experimental and control. Instead, these groups were sort of selected by nature, um, and the groups are the MZ and the DZ twin pairs, right? Causal factor MZ, genetic identity, DZ, um, only partial genetic identity, right? So MZ has the causal factor, DZ does not. All subjects were free of the effect at birth, right? They weren't born with schizophrenia, and then they did use objective diagnostic criteria, so the diagnoses should be reliable, right? And it should rule out some sort of biases that might be introduced by using the objective. And they did sometimes, it says, use follow-up interviews, right, to make sure that the pairs were in fact concordant. Okay, so random sampling. How well does this uh, study approximate a random sampling model? None of them did random sampling, right? Like of all adults in the relevant countries, right? They didn't even randomly sample all twins, right? Instead, they looked up in a database of psychiatric inpatient admissions, right, found schizophrenic people that had twins, right? Um, and then so this is perspective, right? And we're going to explain in a moment what makes it perspective, even though kind of in time, right, they were looking at the effect variable, schizophrenia, and then trying to um, match them to their twins. Um, but at any rate, it was not random, right? Is the point here. Right? So they did rule out, right, as we saw, some potential alternate causal factors, or, for example, right, dizygotic pairs that had one male and one female gender. They ruled out um, pairs where the one of the twins were one member of the pair died early, right? So we weren't uh, diagnosed yet, right? But they couldn't have been, so we wouldn't want that to count as a sort of a not concordant pair. Those got uh, kicked out as well. So they did pay attention, right, to potential failures of randomness, even though they couldn't do a complete random sampling model. Okay, so how do we evaluate this? Okay, well, samples of 180 and 174 for the MZ and DZ twin pairs, right? So um, margin of error no more than 10%, probably a little bit less actually, at 95% confidence. So the difference were pretty big, right? It was between like 47% to 4%. So a 10% margin of error is not going to create any overlap for sure. And we can even do a quick test of the effectiveness in the population, and it'll come out between 0.33 and 0.61. I won't pause. This is going to take so long, this lecture, that I'm not going to pause to do the math for you. You can discuss that in section if you want, how you come to that effectiveness. But okay, so summary. So what do we think, right? This is a combined, right? So it's 5 different twin studies all combined. And given step 5, our evaluation, right, it's definitely statistically significant and appears to be fairly effective, right? And so we can say it's a good example of a prospective design. The diagnostic criteria were objective. Um, the use of follow-up interviews where possible. They used decent variety of geographical locations. Um, they did. There are worries, right? We talked about this assumption that there's environmental similarity when, in fact, growing up with an identical twin is probably fairly different from growing up with a fraternal twin, right? But unless there's some indication that that's known to be a causal factor, right? We maybe don't need to worry about it too much. So it appears that, yeah, inheritance of genetic material is an important causal factor for schizophrenia. This is strong evidence. And here we have the numbers. So this is confusing at first because you might think they're doing percentages, right? But this is 85 out of 180 in the experimental group, right? The monozygotic twins, concordant pairs, and then 7 out of 174 in the control group, uh, which is the DZ. Only 7 concordant pairs among the. Okay, so again, a lot being covered, I understand, in this lecture. Um, do reread the material if you're having trouble. We are going to discuss this on Monday. We'll have a review session, and of course, always come to office hours as well. Um, don't try not to get lost in the details, right? And I really want you to just catch the big picture stuff. I'm not going to be nickel and diming you on like little tiny details on the exam. That's not the point. I want you to get the big picture. Okay, so moving on. Uh, Another study, right? Study about um, if we can get a bit more fine grained on what is, so if schizophrenia is a genetic, uh, right, is a genetic disease, right, or it seems to be inherited, um, can we, is there anything else we can find out about the sort of specific genetic mechanisms involved. Um, this can be hard, right? Particularly if there's several interacting mechanisms that are all involved. Well, we do have techniques now for sort of scanning the entire genomes, like right, of someone's uh, chromosomes and finding locations for potential genetic causal factors. We won't get too technical here; it's even over my head, that right? But one such technique identified one particular sort of gene called DTN-BP1, located on chromosome 6, as a potential cause. Um, so why I think it's a cause? I mean, several reasons, right? It's involved in production of proteins in muscle and brain tissue, and disruption of that process is a form associated with some forms of muscular dystrophy, and one of those has similar symptoms to schizophrenia. Um, there's also evidence that this protein is involved in the production of glutamate, which is a neurotransmitter that is associated with schizophrenia when it's abnormal. So that combined with this sort of like scanning technique, right, which is more a statistical technique, suggests that this may be something to look at as potential causal factors. This particular gene. So we can't do a prospective design here, right? Because there's just not widespread screening for this particular gene in people. Um, and then schizophrenia is not diagnosed until much later in life, right? Um, but there is another sort of study that you can do, and in fact, one was done in 2004, right? That does provide evidence for a positive causal association between this gene and schizophrenia. So they sampled 1,097 adults, 524 cases of schizophrenia, right, without substance abuse, schizophrenia or schizoaffective disorder, slightly different diagnoses. Um, and they made sure to choose people without a history of substance abuse using DSM-4, and then 573 controls. And the controls were themselves screened for any history of psychiatric disorder or substance abuse, right, to make sure that they are, in fact, controls. They used a diverse sample of different genders and ethnicities. Right? So the subjects were all tested for 7 single nucleotide polymorphisms, right? We call them SNPs, that have been identified in earlier studies of schizophrenia. So these are just single like mutations, changes in the genome, right? If you remember all the way back to the beginning, the story of DNA, right? We have these bases, cysteine, thymine were 2 of them, right? So sometimes, right, usually, right, say there's a C in that spot, but it's sometimes replaced by a T. Um, those are the sorts of changes, like mutations that we're looking for. Um, so in that situation, we call the C the major allele, which is the more common one, and then T being the minor allele. So the idea is we want to see, right, if those polymorphisms are associated with schizophrenia. So here it's divided by ethnicity, right? So we've got groups of white people, Hispanic, African American, white and Hispanic combined. Um, we have the different mutations that we're interested in, the polymorphisms that we're interested in, 3, right? For each, you see they're repeated each there. And then we have um, patients with either schizophrenia or schizoaffective disorder, and then patients just with schizophrenia in the second column, right? And we'll see controls, what percentage of the controls had that polymorphism, and what percentage of the cases, meaning people with the diagnosis of schizophrenia or schizoaffective disorder, what percentage of them had that particular polymorphism, right? And you can see kind of looking at it, so just look at that sort of the column where it says patients with schizophrenia or schizoaffective disorder. The controls, the percentages tend to be lower. Okay, so the data is, right, the frequency of, again, that polymorphism was 7% among the combined white and Hispanic controls, and it was 13% in the cases. So higher percentage among cases of schizophrenia or schizoaffective disorder. Is it statistically significant? A little bit harder to eyeball there, right? The difference between 7% and 13% is a little smaller than we were looking at in the last case, where it was like 47% versus 4%, right? It's going to depend on the sample size, stuff like that. But that is an 86% increase, right, from 7% to 13%. So worth looking into. If you limit the sample to just schizophrenics, that is, take out the people with schizoaffective disorder, the frequency goes down from 13% to 12%, right? But that's still a 71% increase compared to controls, so pretty good. There was not actually any difference in African American subjects whatsoever, which is interesting, right? And they sort of like brush over it and they just say, okay, well, our hypothesis is just going to be about white and Hispanic people, right? But very interesting question as to why, right? They say maybe they didn't sample enough African Americans, or it might just be that for whatever reason, that gene is not a cause of schizophrenia in African Americans. Again, okay, but the researchers do conclude, right, that there is evidence that DTNBP1 is a positive causal factor in schizophrenia. So what sort of design was this? What are we looking at here? This is called a retrospective design, right? What's retrospective about it? It started beginning with a sample of a population that already exhibits the effect, right? Schizophrenia, and it looks back to see who has the causal factor. So there's no random selection of subjects. Right? They pick one hospital, they look at every person there with a diagnosed schizophrenia, and they look back and see if they have these mutations, right? And they compare them to people without schizophrenia, see if there's a difference. So why do they need to do this? Why just focus in on people with the effect? Well, if you tried to randomly sample just the whole population, you're not going to get enough people with schizophrenia. You would have to sample like 100,000 people. Remember, it's a 1% of the population tends to have it, right? So it's just not practical to sample that many people. So they have to start off, find people with schizophrenia, and then work backwards with retrospective design. So you do have to look out for potential bias, right? But they did, again, use objective criteria. These are people diagnosed using the DSM, right, in a clinical setting. There can be other ways that bias can creep in, though. So, for example, people could choose whether to participate in this study or not, right? It was voluntary. Well, is there some systematic difference between people who choose to participate in such studies and people who don't, right? And are people who would choose to participate in such a study, would be they somehow more likely to have schizophrenia or not, or to have the mutations of these genes or not, rather? Sorry. Who knows, right? But you have to look at all these possibilities, right? Another potential difference, right? You're sampling from a hospital, right? So that means you're sampling from people that can afford to go to a hospital or have some kind of health insurance. That's going to correlate with higher socioeconomic status, depending where you're in, what country you're in, and whatnot. Some countries have universal health care. It's not an issue. But these are all things you have to think about. Stuff you don't have to think about when you're using a random experimental design. So, as the case with prospective designs, you have to be careful to control for any possible alternate causal variables. For example, right, that in the sample, there's significantly more males than females, right? So if it turned out that being male is a causal factor in developing schizophrenia, or even just has an interaction effect with the actual causal factor, that would be a problem, right? So when we look in a moment at figure 8.9, this hopefully should clarify the difference between retrospective design and a prospective. In a retrospective design, the population you're sampling is the population with the effect, right? And then you're looking backwards, see, right, the observed relevant frequencies of the proposed causal factor in that population. Exactly the reverse of a prospective design, right? So you may recall with our twin studies, right, we were looking at the causal factors, genetic identity and genetic diversity, and then seeing within those groups, right, what are the relative frequencies of the effects. So look at this chart here, right? So here we have the population sampled, right, which is all people with schizophrenia, and then you have your control group, right? And then we're looking to see in each of these groups, uh, what is the relative frequency of the causal factor. So when I say conceptually, it's the reverse of the perspective. This is what makes something perspective. Okay, so in many ways, retrospective studies are actually quite simpler to survey sampling, right? So, and at least in principle, all of the information you get from a retrospective study could be obtained by a survey, right? So instead of matching a control group and experimental group, you could just do a random sample of the population, compare the percentage of S&P variance in schizophrenics to the percentage in others, right? Non-schizophrenics. So why not just do samples? The problem I've discussed already, it's numbers, right? So the rate of schizophrenia in the population is 1%. So you'd need a sample of 10,000 people to hopefully find 100 cases of schizophrenia, and you'd still have a margin of error of 10%, which is a bit more than you'd like. So really, to get a good, nice, tight margin of error, you'd want to sample 100,000 people, and that's just really hard to do and very expensive. So ways that bias can creep in. We already mentioned sort of non-response bias. So not everybody's, it's a voluntary study. Not everyone's going to choose to participate. And maybe there's some systematic difference between people who would choose to participate and those who would not, right? Other kinds of biases include surveillance bias. So that's something that happens when you're being observed, right? So as an example of that, in the 80s, in the UK, they showed that long-term use of birth control pills positive causal factor for breast cancer, right? Um, but it turns out, problem with that study is women who take birth controls, they go to the doctor. That's how you get prescribed birth controls from your control pills like at the time, right? Was from your doctor. If you go to the doctor more, you're more likely to have your breast cancer detected, right? Just because you're a person who is on top of your right healthcare or whatever. Um, so ultimately, they decided like that bias didn't impact the conclusion of the study, but it's the sort of thing you have to look out for, right? Um, so there's not much of a chance of surveillance bias here, right? So none of the subjects, typically you don't get tested for those particular genetic mutations, right? So it's very unlikely that any of the subjects would have been aware that they had the mutation and that was somehow affecting their behavior. Um, having, if you don't know that you have the mutation, it's not plausible at all that it would somehow make it more likely that you get diagnosed. Not making you go to the doctor more or anything. Other types of bias include what we call recall bias. So sometimes studies will rely on the subject's memory, right? So for example, in the birth control breast cancer study, so say a woman's diagnosed with breast cancer, and then you ask them if they had taken birth control or not, sort of a retrospective design. The fact that you were diagnosed with breast cancer could unconsciously make you report taking birth control more than you had before if you suspect that it's a causal factor or it's just somehow salient to you, right? So to avoid this sort of bias, you'll get professional interviews that are sort of good. This is their job, is to use very standard techniques and avoid those sorts of biases. Um, right, it turned out, for example, in that birth control group that everybody actually overestimated the amount of time they'd been on birth control. So there was a bias, but it sort of balanced out in both experimental and control. But in this study, they're not relying on anybody's memory. They're using medical records, so it doesn't seem like recall bias would be relevant. You can also have interviewer bias. So you can't always, obviously you want a double-blind study, right? So even the people that are the subjects don't know if they're experimental or control, and then um, ideally the researchers don't know either, but sometimes it's impossible to keep the interviewers totally in the dark. Um, and when they are aware of some of these details, it can introduce bias. So you have to have very standardized interview procedures. And in this study, it seems like they did, right? The only interviews involved were these assessments of schizophrenia were done by trained professionals using standardized criteria. Um, the data for the genetic mutations was obtained from blood samples. So it doesn't seem like too much chance of interviewer bias in this case. Um, we discussed many of these same biases when we talked about survey sampling, right? And techniques for avoiding them in the same in other cases. So the effectiveness of a causal factor is that something you can measure in retrospective studies? Well, recall the effectiveness of a causal factor, we defined it as the percentage of the population that would exhibit the effect if all or none of them possess the causal factor. Um, Now, a prospective study does just that. It takes two groups, one where all the individuals have the causal factor, right? They all have genetics identity, and one where they don't, right? The dizygotic twins. Um, and then you determine whether there's a difference, right, in the frequency of the effect between the two groups. A retrospective study can't do this, unfortunately. Right? So knowing that 13% of white and Hispanic schizophrenics have the relevant, right, SNP, it's just not the same thing. So you can't use retrospective studies alone to estimate effectiveness of a causal factor. Um, some studies will use other information to do this, but that's sort of beyond um, anything we're going to study in this course, right? So if the study is published in a reputable journal and they say something about the effectiveness, you can trust that, right? The peer review process, uh, they checked out, right, their whatever methods they used, and it's got to be true, or at least evidence. Okay, so how do we evaluate retrospective studies then? And I know we're over 50 minutes at this point. We're closing in. We got like 5 slides left. Uh, Basic strategy is going to be uh, basically the same as evaluating experimental and prospective studies, except for the whole design is reversed, right? So we're looking for a statistically significant difference between the frequency of the cause in the experimental and control group, right, as opposed to effect. If there is a difference there, then that's evidence of a causal connection between the cause variable and the effect variable in the population. Now, the main trouble you have to look out for in retrospective studies, you just can't get random sampling. So you have to focus in, take a lot of time looking for potential sources of bias, right? It's going to be also pretty difficult to rule out alternative causal factors. Um, So because of all the problems associated with these retrospective studies, as a general rule of thumb, if you've identified that you're dealing with a retrospective design, you shouldn't evaluate it as any better than causal hypothesis is moderately well supported, right? Even the best retrospective study, just go with moderately well supported. But that doesn't mean that they're worthless, right? They just, one great thing they can do is give you, they're relatively easy to conduct compared to some of these other studies. And if you get right some moderate support from one of these studies, that's a good reason to investigate further, right, and do hopefully other types of designs to investigate that causal hypothesis. Okay, so last thing we're going to do is just evaluate this DTN-BP1 schizophrenia study. So this is, as it's presented in the book, again, not expecting you to do anything this exhaustive, but I want to give you the whole exhaustive thing because it'll hopefully make sense to you, right? So step one, what is the real world population? What's the causal hypothesis? Well, the population of interest was adults of white and Hispanic ethnic origin, uh, and the population actually sampled consisted of white and Hispanic cases, right? So by cases, again, we meant people diagnosed with schizophrenia or schizoaffective disorder, um, and some controls, right, with no history of psychiatric disorders or in their family and no drug abuse. So both males and females were included. Um, they all were recruited from inpatient and outpatient hospital admissions. Uh, so, I mean, roughly representative U.S. adults, like a lot of us, you know, go to hospitals for various reasons. But adults living in rural communities where there aren't any hospitals, or people that don't have health insurance and avoid hospitals, they probably wouldn't have been sampled. So that could be an issue. What's the cause variable? It's the NBP1 gene, right, which is marked by the presence of this particular uh, polymorphism, right, P1578 SNP. The effect variable, schizophrenia or schizoaffective disorder, as diagnosed by the DSM-4. The causal hypothesis is that uh, activity controlled by the DTNBP1 gene is a positive causal factor. All right, step two, what is the data? Well, of 309 cases, white or Hispanics with schizophrenia, again, the cases means you had schizophrenia or schizoaffective disorder. 13% of those cases had the polymorphism, and of the 499 controls, 7% had the polymorphism. When you limited it to only schizophrenia, it was 12%, right? Had of the cases had the polymorphism. Okay, so what is the design of the experiment? It's you should have identified it as a retrospective design. Notice in step one, right? What was the population sampled? People with schizophrenia, right? So, and that is the effect variable. So this is a retrospective design. The control group was constructed, right, by matching for other characteristics of the members of the experiment. Found people at the same hospital, presumably similar demographics, right? So that was your control group. So does this approximate a random sampling model? Well, it certainly was not random, right? They picked people with the effect. But they did consider, right, some potentially confounding factors. So they did rule out anybody with drug abuse. The controls were screened to make sure that none of them had any history of psychiatric disorders or immediate family members, and so on. They did use, right, diagnoses obtained in clinical, right, situations by trained people using the DSM-4 criteria. Um, the testing for the genetic markers was objective, just using blood samples. Uh, so they didn't have to deal with a lot of these various biases like surveillance bias, interviewer bias. So that's pretty good, right? It's in a retrospective design, you're doing a lot to compensate for the lack of random sampling. So altogether, a moderately good fit. You're never going to get a great fit to a random sampling model in a retrospective design, but so moderately good is as good as you're going to get, and good as studies could probably get. Step five, evaluating the hypothesis. So, um, assuming a random sampling model, there is, they claim, and certainly in this actual study, if you read the study, right, start off with what they report. They report a statistically significant difference, right? Now, notice when we do the math, it's kind of close, actually, right? So the margin of error for a sample of 618 is 3 to 4%, right? The margin of error for a sample of 998, and again, so notice this is alleles, so it's like double the number of people, right? Because each you have two different sort of like two different alleles, right? Genetic markers that they're. So so that's 3%. So the sum uh, is around 6 to 7%. That's your margin of error, like the sum of the margins of error. And the difference in the frequencies, 13% minus 7%, is right around there, right? So like you might be tempted to say they overlap, right? Um, but since the study did more sophisticated statistical techniques and they say that's statistically significant, then go with what the study says, right? We're seeing it's close, right? And if they used a little more sophisticated statistics than we did, then we'll believe them. They're not from our rough math. They're not totally off, so it's totally okay. So yeah, it looks like there's support, right, for the hypothesis, um, and we can't estimate the effectiveness because it's a red. Okay, last slide. So we're at an hour. I went about 10 minutes over, so apologies. Um, but this is the only lecture you have to watch this week because we had a holiday Monday, so that makes a little bit. Um, so summarizing, right? This is a retrospective study. We know problems with retrospective studies in general, but as those go, it's a good one. They used objective criteria throughout, right, to group the cases, to test for the causal factor, um, and they did their best to rule out some common types of biases. Uh, so they really did as well as they could to try to compensate for the lack of random sampling. So we can say, again, but because it's retrospective, at best, we can say moderately strong evidence, right, that the causal hypothesis is correct. And taken in conjunction with other recent studies, um, we didn't look at those, so they mentioned those. I don't remember why, but again, you wouldn't be expected to put all of this in this. We'll talk more about this on Monday in our review of sort of more like what's expected of you on the exam. I understand that what's being covered in this exam is a lot. I'm going to try to like meet you halfway here. I'm not trying to mess anybody up. Okay, so I will stop talking. You've been listening to me for long enough, and yeah, I will see you on Monday.